Just wanted to start today, since there haven't been industrial speakers in this meeting before, with a little bit of a perspective on why do we care about CH functionalization in industry and really organic synthesis in general. Why are new methods so critical in the work that we do to treat human disease? So to start, rather than doing my own presentation, I thought I would just highlight a couple of papers on the subject. So Lipinski and Hopkins at Pfizer back in 2004 now really just nicely described chemical space for biology and medicine in the context of drug-like chemical space, really highlighting the fact that we've only just barely scratched the surface of all the the potential compounds that fit in this drug-like space, and we really need new methods to access this. And so despite this call, I guess 14 years ago now, just 2 years ago, this paper was published from AstraZeneca, basically highlighting the fact that still a lot of discovery chemists are using primarily just amide bond forming and Suzuki cross-coupling reactions and not much else. And as they highlighted in this title, where have all the new reactions gone? So really using this limited set of reactions leads to this narrowly populated chemical space. So just a few months ago, I think there's been a lot of efforts in this area over the last few years. And a few months ago, this nice Nature Chemistry paper appeared from people at 5 different pharma companies, really highlighting the fact that organic synthesis remains a rate-limiting factor in drug discovery, and that really exploring non-traditional disconnections, including CH functionalization, this is critical to be able to access some of this not yet explored chemical space that's out there. They also highlighted nicely the critical role that academic research and academic collaborations plays in this, which I'll definitely highlight at the end of my talk today. Tim Cernak and Shane Kriska and a couple others from Merck really described nicely in this review the impact of CH functionalization for late-stage functionalization of pharmaceuticals. So with that, just I wanted to highlight the importance of the CCHF collaboration at AbbVie in this regard. So this nice review was published last year by Hugh and Dan Morton on the collective approach to CH functionalization that's been presented to us and other companies. To be able to tap into some of the wealth of knowledge and expertise in this field. We've had benefited from being able to access and really network with, collaborate with many professors at universities across the country and across the world, and enjoyed many ongoing benefits, including one that I really wanted to highlight, which is hiring top-tier candidates. So over the last few years, we've actually hired, I guess, 8 different candidates from 7 different research groups at the CCHF. So now, when we're interested in a method, we don't just, we actually can just walk down the hall or walk, down a couple of lab bays and talk to someone who's actually had firsthand experience with this chemistry. So I think that's been a real benefit of engagement with the center. So to get into the heart of my talk today, I'm going to talk about as comprehensively as possible about the different kinds of CH functionalization that have been utilized at AbbVie. So as is the case probably across the industry, CH borylation, iridium catalyzed Hartwig CH borylation has been widely utilized. So I'll go over specifically examples of heterocycles that have worked well and not as well in this chemistry. I'll then focus on CH aerylation, talking about some project impact there and some unpublished work from my group in a specific project, TRIP-A1. Finally, I'll move on to what has been less broadly utilized, and I think there's still a lot of room for development in the area of sp3-CH functionalization. I'll talk about a couple of examples from AbbVie, and then several academic collaborations that we've been involved with to further explore this important area of research. So to start with CH borylation, a number of monocyclic heterocycles have this published chemistry and some unpublished. Certainly, some well-established and published methods for diborylation of simple furan and pyrrole have been carried out in multi-gram scale, as well as substituted furans and pyrazoles and 1-pod borylation Suzuki's. These methods have all been utilized broadly, including these examples I was able to show today, including some slightly more elaborate ones containing tertiary amines, undergoing this iridium catalyzed CH borylation from Hartwig quite nicely. Those who carried out this chemistry shown here in the bottom of the slide. Looking at indoles, CH borylation has also been very broadly utilized on multi-gram scale. So a number of substitute indoles are shown here with the results on multi-gram scale, quite good. Alexa and Smith published on the regiochemistry of these substrates and others in this paper. One clear limitation here, sometimes with the right heterocycle, you can certainly present challenges with this most established CH functionalization chemistry. So, for example, this sulfonyl substituted pyridine appears to be somewhat complicating the CH correlation, inhibiting the reaction and limiting us to about 30% conversion in this sort of regioselective CH correlation under conditions that were available at the time this was done. Looking at indole-related heterocycles, additional nitrogens, aromatic rings, sort of regioisomeric aza indoles undergo this chemistry quite nicely, and have been explored in the context of multiple projects, and this has worked quite well. Along the lines of rules shown by Hartwig in this Jack's paper. However, there are cases where functionality such as this pyridone certainly complicates the reaction and inhibits the catalyst to some extent. However, with this specific example, I'll show you in a couple of slides a nice direct CH aerylation solution to this that circumvented this challenging CH correlation. So with that, moving on to CH aerylation, the other kind of large area of sp2CH functionalization that's been utilized broadly at AbbVie. These are two nice examples that have been published both in the patent literature and the primary literature. The top one, a palladium catalyzed CH aerylation to make these intermediates that could be oxidized and undergo SNAR chemistry to access these imidazole pyrazines as p38 MAP kinase inhibitors for cancer. In this BMCL paper describes this is the solution I mentioned to the problematic CH correlation, a very closely related heterocycle where in this case dihydropyridone does not completely shut down this reaction. Yields still moderate in this case, but on a couple gram scale, this reaction worked reasonably well and allowed, following a couple more steps, access to a wide variety of these BET bromodomain inhibitors. And this was, as I mentioned, published just last year. So some unpublished work. This is a manuscript in preparation from colleagues in Germany. They were kind enough to let me present some of this for a project at AbbVie. We were in need of a very robust copper-free process to access many diverse oxazoles. And this method using palladium acetate and DPE-phos was identified that turned out to be very broad and useful for the purpose that it was needed for in this context. So benzoxazoles as well as a variety of substituted oxazoles containing free amines, primary alcohols, were all very well tolerated in this chemistry, and a variety of other heterocycles beyond oxazoles also performed quite well here. Most closely related method was published by Walsh in 2014. However, in our hands, this modified method in toluene at elevated temperature worked quite well, and this will be published later this year. So an example from my group that really got us interested and excited about CH functionalization to begin with, I want to spend a little bit of time on. So we were working on a TRPA1 antagonist project for pain. And the original route to this TRPA1 antagonist 1 is shown here. In the interest of time, I'm not going to go over this in detail, but needless to say, there was many steps that it took to access this key bromothiophenene intermediate fused to a pyrimidine dione. Suzuki cross-coupling then proceeded without any issues, and ester to amide followed by Buchwald-Hartwig CN bond formation gave the final compound 1. Unfortunately, this was rather lengthy, and we needed rapid access to this compound on multigram scale for advanced preclinical characterization. So we were able to, first we did look at other methods to access this bromide more efficiently, and those were quite challenging. However, we were able to access this, the parent thiophene in only one step, and from here investigated a number of thiophene CH functionalization methods. The first ones we looked at were published by Itami several years ago. We looked at both aryl boronic acid and aryl iodide direct CH aerylation under conditions published in these papers. Unfortunately, the boronic acid reaction gave a 1-to-1 rejoisomer ratio of the desired 3 substituted to the 2 substituted isomer with a lot of remaining starting material. And the iodide, while giving a slightly better ratio of A to B, 2-to-1 ratio, conversion was actually worse, and the profile was significantly messier. However, looking at this aryl silane CH functionalization gave a very clean profile under these conditions, published in 2012, giving us a good yield of an intermediate together with some carboxylic acid that could also be used on a gram scale. And then a direct ester to amide exchange gave TRPA1 antagonist 1 in only 3 total steps. This was a nice advance that allowed us to access quantities of this material for advanced characterization. So, as you see, when we've widely utilized, sp2-CH functionalization at AbbVie, and also have been very interested in and have many academic collaborations involving sp3-CH functionalization. I do want to share a couple of project-related examples. I can't show the specific structures that have been looked at, but these are figures from these papers. This one from Jin-Kwan Yu, this one from Chris Kramer, and Nikolai Kramer. So this Jin-Kwan Yu method for pyrrolidine CH aerylation was utilized broadly with these examples, some of these examples from the paper and some others. However, there were some limitations here, specifically quinolines, indazoles, very sterically hindered aryl boronic acid, as well as some more hindered pyrrolidines were all somewhat problematic in this chemistry. So that was something that was a limitation here. And this specific reaction that's published in this paper, actually in Kramer's paper, actually worked very well. Instead of using par-methoxybenzyl, using trimethoxybenzyl in order to more easily remove the protecting group. This was scaled to 10 grams under slightly modified conditions using less palladium and ligand, giving an 85% yield and 95% EE. Mark Medalenko and my group carried this out, and this worked really well to access a lead compound for a project just last year. So with that, I'd like to focus now on academic collaborations that we've really sought to further explore sp3-CH functionalization and probe its utility in the pharmaceutically relevant sort of arena. So wanted to start with our collaboration with Hugh's group at Emory. So a few years ago, we were really enamored with this method. Davies and Morton published this review looking at these donor acceptor diazo compounds doing CH functionalization into many different sp3-CH bonds. Ones that we were particularly interested in were small saturated heterocycles such as CHF and pyrrolidine. A few years later, using a new class of catalysts, these BP-CP catalysts, Hugh and co-workers showed that actually using trichloroethyl ester diazos, you could do really interesting CH functionalization into methyl groups. And while this was very interesting, perhaps more interesting to us was the fact that these conditions in dichloromethane with the trichloroethyl esters allowed for the first time utilization of some heterocyclic diazos that would engage in this chemistry. So while the yields and the anti-selectivities weren't always as good as the simpler aryl system, this was significantly better than the original systems, and we thought this was really worth further exploration and collaboration. So where we wanted to apply this was in the area of fragment-based drug discovery. So we recently have an approved drug in oncology, venetoclax, that is really a success story for fragment-based drug discovery. This review a couple years ago describes the discovery of this drug using fragment-based drug discovery. It was a 20-year journey that recently culminated in the approval of this drug. Really exciting story. And so this is really all about taking small non-lipophilic fragments and looking for weak binding and then optimizing as you build up sort of a stronger binding drug-like compound. So I'm not going to go over these in detail, but in general, small and non-lipophilic compounds are of interest. So that's where these sort of heterocycles and not too many aromatic rings really becomes important. So we saw this Davies chemistry as an opportunity to really benefit our fragment-based drug discovery efforts. So in this collaboration, we've had a number of directions that we've looked at. The first, we really wanted to be able to access more of these trichloroethyl esters. The existing diazo transfer chemistry was somewhat limiting, so we were really interested in this method by Wong and coworkers. There was a CH functionalization itself with cross-coupling with aryl iodides, slightly modified conditions with trichloroethyl esters, including triethylphosphine, allowed the coupling of numerous aryl iodides, including heterocyclic examples shown here. And this really allowed access to many compounds that were previously more difficult to access with other available methods. So with this, we've been looking at many interesting CH functionalization reactions of small saturated heterocycles, including these silocyclobutanes and pentanes, as well as several others that aren't yet published. This was just published very recently. So another thing that we've explored is this paper in Nature, published by Hugh Davies and Quangbao Liao, who's now actually an AbbVie colleague. We're really interested in this Suzuki cross-coupling to access a wide variety of rhodium catalysts. The interesting thing here is subtle changes in this catalyst architecture cause dramatic, remarkable differences in selectivity profiles for these CH functionalization reactions. In this paper specifically, this methylene CH functionalization was being explored. A library of catalysts using this one-step Suzuki cross-coupling library method identified this specific catalyst 15 that was uniquely effective for this transformation. And so we thought, well, why not, make a much larger library of catalysts? And since we have a very, excellent high throughput chemistry capabilities here, Quangbao visited AbbVie for a summer and was able to make many more catalysts and explore the utility, which was a productive visit here. And also, a chemist from AbbVie was able to visit Emory and really work on expanding the practicality and scalability of some of Hugh's chemistry for application in the pharmaceutical industry. So moving on to other academic collaborations, we had a collaboration with Melanie Sanford's group at Michigan that really sought to further explore this very interesting transannular CH functionalization of this 310 bicyclic ring system. The paper describing this work and improved deprotection conditions will hopefully be published very soon. And this was to benefit our fragment-based drug discovery our efforts. This paper was published earlier this year, collaboration with Tim Noel's group. An interesting flow oxidative tungsten catalyzed photochemical process that is a new method to access some carbonyl compounds from methylenes, including some examples with pyridines and more elaborately substituted or more complex substrates that are shown here. Also, collaboration with Ian Baxendale at Durham University has explored accessing some highly functionalized azidines via CH functionalization. And then the last academic collaboration I wanted to highlight is that with Shannon Stahl at UW-Madison, where we've been looking at this alpha cyanation of cyclic secondary amines using some electrochemical methods. These can get to 2-cyanoamines and unnatural amino acids. A manuscript describing this work is in progress and will hopefully be out shortly. And we're in the process of looking into 2 additional CCHF collaborations as well to further explore sp3-CH functionalization. So in the interest of time, I'll make a few concluding statements here. Of course, this isn't comprehensive. I just tried to give you a pretty broad overview of a kind of CH functionalization that we're looking at AbbVie. Clearly organic synthesis, as I hope appreciated, continues to drive pharmaceutical discovery and development. New methodologies such as CH functionalization are really critical for us to find therapeutics for the world's deadliest diseases. Definitely, sp2-CH functionalization is currently more developed and broadly implemented than sp3-CH functionalization, but I think that's changing. But it certainly remains a challenge to further expand the functional group and heterocycle compatibility of sp3-CH functionalization methods. I'll just throw out a few hypothetical challenges here. This 310 system, there's existing methods for this methylene, for this methylene. However, this methine, there's not yet a method for that. So ring systems such as Chroman, while we can definitely, CH functionalize the benzylic position, these other positions are not yet accessible with existing methods. And if you put a couple extra heteroatoms in there, this becomes an even more challenging problem. With perhaps a methyl group off of a Chroman like this. So there's certainly many methods that have been used at AbbVie, but I either couldn't discuss or didn't have the time to discuss. Many of them are shown here. We actually have some very interesting biocatalytic results on regioselective late-stage oxidation chemistry that will hopefully be published soon. And we're actually even considering a commercial route to a drug that uses a direct CH aerylation. Finally, there's a book chapter in progress in the next edition of Practical Synthetic Organic Chemistry. This will, for the first time, include a chapter on selective metal-mediated CH functionalization. So my colleague Shashank Shakar is drafting this chapter. So be on the lookout for that book. It will be out shortly. Finally, I'd just like to acknowledge my group, past and present. This is a picture of the group from earlier this year, kind of fun outing that we had. AbbVie is a great place to work. So I'd like to also thank all the AbbVie chemists in Germany and Ludwigshafen in Germany and Worcester, Massachusetts, and here in Lake County, our process and discovery chemists who've used CH functionalization in their research, championed the collaborations that I described, and allowed me to present their work today. I showed their names on the slides when I presented their work, and thank you very much again for the honor and opportunity to present today.